Hey class, Dr. Frizzell here. We're going to be talking a little bit about what neuroscience can do for us. In these slides, we're going to go over a few basic concepts, and hopefully I can provide a little bit of background in these areas to kind of help maybe provide a little better understanding of what these different areas are and what kind of challenges we face as a discipline and a science. In the first area, under neuropsychology, this is an area that is near and dear to my heart. I spent a couple of years in graduate school doing this kind of work for a university hospital, and it was absolutely fascinating to see real life cases in which patients had lesions in the brain. And a lesion is essentially another word for some type of focused damage to the actual brain tissue, and that can be due to a degeneration of the cells. It could be due to physical damage, like from a gunshot wound or a penetrating head injury. Sometimes that's from an internal head injury, like from a concussion or just getting hit in the head really hard and causing brain damage that way. Sometimes it's a biological anomaly like hydrocephalus. There's all kinds of different things that can cause lesions in the brain. So what we try to do in neuropsychology is we try to understand what happens to a person's behavior, what happens to their emotions, what happens to their ability to think when they have lesions in very specific areas of the brain. In the long-term history, we had difficulty being able to do this because we didn't have imaging studies like we do now, like CT scans, MRIs, functional MRIs, PET scans, things like that. We didn't have those, maybe 100 years ago or so. And so what people had to do who were studying this was they would see people that had specific types of psychological problems that we believed were the result of a lesion in the brain, and we had to essentially wait for them to die. And then after they passed away, they would take a look at the brain and try to figure out where the damage was, and then make a correlation across multiple cases where people had similar types of behavior problems, and then look and see if there were any consistencies as to where the lesions were in the brain. So best case scenario, what we would see is, a person that has difficulty with executive function, meaning they had difficulty paying attention to more than one task, they had difficulty making decisions and organizing information. We would expect to see some type of lesion in the frontal lobe. Ideally, that's what we would see, but as you can see on this slide, some of the challenges include that naturally occurring lesions are never very clean, and they're never in a specific area of the brain where we can just sort of say, oh, that's what it is, and hone in on one specific little tiny piece of damage. Usually what happens is there is a large amount of damage that covers several different areas of the brain at once, and so we see more than one type of damage and more than one type of behavioral problem at the same time. And in addition to that, the pathways for different functions are so closely tied together and interlinked that sometimes when we see a lesion in the brain, it can be very small, but it can affect more than one behavioral issue or psychological issue at one time. So it can get really, really difficult to sort of figure out what is going on when there is a small amount of damage to the brain, what's actually being affected. As you can see in points number 2 and 3, behavioral profiles and specific deficits aren't always a clean correlation with each other. Sometimes we can have a lesion to an area in the optic nerve area, and that could be the reason why someone is having a visual deficit. On the other hand, they could have some type of neurodegeneration in the occipital lobe where the visual processing cortex is, and that could also explain it. So Sometimes it can be really difficult to figure out where this is coming from, and if a person has more than one lesion, then we really have difficulty figuring out what part of the brain is responsible for a specific deficit. In some cases, as you can see under point number 4, human brains are different from person to person, and we often hear the phrase that everybody's wired a little bit different. Well, that is both not true and true at the same time, because even though everyone has a similar brain structure for the most part, that is, unless they have some type of congenital defect or massive brain damage, generally speaking, people have similar brains, but that doesn't mean that the connections that each person have are going to be the same from person to person. For example, there may be some people whose auditory connections are completely miswired. There may be someone who has very strong visual pathways, and so their visual acuity and visual memory and so forth are very, very strong. In fact, they might be better than the average person. So this lends itself to lots of different kinds of strengths and weaknesses that people have, but it makes it difficult when we look at how lesions to a specific area might affect one person one way, but can affect another person in a different way. Looking at point number 5, where it says each function is served by a network of brain areas and not only one area in particular, the way that you can kind of think of this is when you're on your laptop or mobile device and your internet connection goes down. What is the cause of that? Well, you could have a problem with your phone or your computer. There could be a problem with your physical, router or the actual network, or the internet service provider could be having problems. There might be some type of electromagnetic interference. You might have some other issue going on, like a virus or something that's interfering. There's so many different explanations because all of these things are interconnected in some way. And the same thing is true for the brain. Many of the functions that occur in the brain are interconnected with each other, and they're interconnected so microscopically, and it's so difficult to tease all of that apart that sometimes it can be difficult to know where in the network something is going wrong. As a science, we are still learning so much about what the brain does and what it can do. This science is always evolving and we're still progressing. And so what we think we know in 2020 about what the brain can and cannot do may be completely different than what we know in 2025 or 2035 or 2045. So we may come to some huge revelations about how to solve major problems like Alzheimer's disease, brain damage, treating concussions, and so forth, based on that understanding. As of now, we still have a fairly limited understanding, but we are improving every day. So in this slide, we're going to look at a brain scan of an individual who has some damage there. You can see in the lower left quadrant of the brain, they've got some white hypodensity, which is sort of like a degenerative area of the brain where things are sort of deteriorating and not working very well. And what we can find out about this person is that they have a resulting condition called agnosia. Agnosia is when a person has difficulty interpreting sensations and being able to recognize things. So one of the most common things that we see is being able to name an object. And so we have developed some tests to sort of show people that have lesions in those areas of the brain, and we show them pictures of different objects and they have difficulty naming those objects. And if they make a certain number of mistakes, then we know that they have some pretty significant problems. Occasionally, even everyday people will forget what something is called and look at it and say, I know what the word for that is, but I just can't quite think of it. That's not what we're talking about here. these individuals have difficulty naming even everyday things like a shoe or a pencil or, a cup. So this isn't just limited to being able to name objects. As you can see under number 2, there are specific types of agnosia, like prosopagnosia, which is a form of agnosia, meaning that we have difficulty recognizing people's faces. So there might be some difficulty in recognizing people who who are in your life, like maybe your brother or your significant other. You wouldn't be able to recognize their face. You might be able to recognize their voice, and you may have some familiarity with their personality, but if you saw them walking down the street, you would not know who they were if you had prosopagnosia. Same thing with achromatopsia, which is a difficulty being able to interpret different versions of color, and so on. So as you can see, we can get really deep into the weeds on different types of lesions and specific types of the brain. And like just for visual recognition, there are even specific types of visual recognition problems. And the same thing can be true for being able to interpret auditory information, sensory information on the skin, interpreting smells, and so forth, as we discussed in the other set of slides for this week. This specific person, if you look at where that damage is, they actually have several of these subtypes of agnosia all at the same time. And as you look at that damage there, it is not a small area. That's actually pretty diffuse, spread out across kind of a major subsection of the brain. But it's going to be hard for us to tell which parts of that hypodense white piece in there is responsible for the facial recognition, the color recognition, and other types of visual recognition. However, at the end of the day, the term visual agnosia turns out to be a very broad category, and it covers all these specific things that we were discussing here. And there are multiple types of this, and some are fairly common for people that have specific types of brain damage or stroke challenges, and some are exceptionally rare. They're so rare that maybe only a handful of cases have ever been seen by doctors across the entire span of time. So I would encourage you guys to take a look at this and maybe do a little digging on the internet if you're interested, and find some really interesting anomalies that are due to lesions in the brain that can cause some pretty unusual results. Okay, so what we're looking at here is an fMRI, which is a functional magnetic resonance imaging. And what this does is it uses a series of magnetic waves to essentially draw a picture of the brain, and then at the same time, it can locate where in the brain it is digesting and processing oxygen the most. And they do that for a specific task to see when a person is engaging in a task, what part of the brain is active the most. If you guys think about it, what we find is that the brain is kind of an energy hog. It uses the vast majority of the sugar and oxygen and nutrients that we put into our body. And so it only is logical that we would look for, where in the brain is the energy or nutrients being used up the most during a specific task. And that's exactly what the fMRI does. So what the object is here is we're trying to get an idea of what's going on with this particular individual for gustatory processing, which is the ability to taste things. All the different colored pieces of this fMRI show different researchers who have done different things to sort of try to provoke a person's gustatory processing area. And what you can see is that it's pretty close to the same general area, of the brain. If you look really carefully, what you'll see is that this is in the sort of centralized location of the frontal lobes, more sort of on the bottom facet of the frontal lobes, and sort of on the left and the right at the same time. So what's really interesting about this is that it seems to be kind of diffuse. It's sort of all over the place in that one area. It's not super defined. So what this tells us is that, there might be different kinds of smells that might affect this person slightly differently, and those smells might be activating different parts of the brain at the same time. For example, if they smelled an odor that was a little bit off-putting, it might remind them something and it might activate the memory center of the brain. If they smelled something that they might associate with a trauma, like smoke, for example, that might activate a fear center of the brain. And so we're looking at this, trying to sort of figure out where is the gustatory processing area of the brain. And one of the remaining questions here is, why is it still even spread out as much as it is right here? We can kind of localize where in the brain this is at, but it's not super defined. And when I say it's not super defined, you look at the green part of the text here and it says the fMRI has a spatial resolution of around 1 millimeter. At face value, that sounds like it's pretty defined, but if you consider that 1 millimeter contains thousands, literally, of neurons that could potentially serve different functions and different pathways, then it feels like, maybe the fMRI is telling us something, but it's not getting at this on a highly, highly defined level like we would want it to. And like I said in the previous slide, we're still trying to get there as a science and still making forward progress. It's just we're not quite there yet. So a couple more things about this graphic. One of the things that they're trying to do as well, and I didn't mention this before, is that when they're looking at somebody with gustatory processing, that is, they're trying to discover, where the taste center is in the brain where we process that information. One of the ways that they measure that is they measure the person at baseline, which means they're not getting any kind of stimulus that would cause them to taste something, but they take that information and they see what the brain is doing when there's nothing going on taste-wise, and then they give them a specific flavor or taste, and then they measure where the brain is lighting up, and that's why it says gustatory processing versus baseline. So for now, I think it's probably sufficient to say that there are multiple studies that show that those areas right behind the brain, right behind the eyes, and we call the orbital frontal area, is probably the area that we can hone down and say is as best as we know the gustatory processing area of the brain. One of the other limitations of fMRIs, as highly advanced and as fascinating as they are, is that the fMRI doesn't have the ability to look at activities in the brain that are only milliseconds apart. So there might be 10 different things going on at the same time in this part of the brain, and they're literally only milliseconds apart. And so how do we really know for sure what it is that the brain is doing in that specific area? We know that something is happening, but we can't explain all of it just by looking at this graphic. So I guess the take-home message here is that imaging studies do help us to get some better ideas about what's going on in the brain, about where certain types of sensory information is being processed, but we can't rely solely on these imaging studies. The good news is that we don't have to wait for a person to die who has some type of sensory processing problem to be able to take a look at their brain and figure out where that is. We can actually do this in living individuals who are functioning normally and compare them to people who are having problems. And this adds a lot of depth to the science. However, these imaging studies, as I pointed out in these slides, they are definitely not the be-all, end-all of information. And so They are an important piece of information, but we're still not quite there yet as a science in terms of imaging studies, understanding what the brain can and can't do. Have a great week and tune in for Module 2.